Hello everyone and welcome to our module on genetic principles. In this video I'm going to go through some vocabulary and some basic principles that you need to understand in order to watch the other videos on genetic diseases. So let's start with some basic terminology. The genome is the DNA contained in the nucleus of a eukaryotic cell. It's considered the hereditary material that can be passed on to successive generations of cells. Genes are portions of the DNA or genome inside a nucleus of a cell, and they code for proteins that carry out specific functions. A chromosome is a rod-shaped cellular organelle. You can see these under the microscope. I'll show you a picture of chromosomes in a minute. And a chromosome contains a single continuous DNA double helix strand, which contains a collection of our genes or DNA. And human cells have 46 chromosomes arranged in 23 pairs. The first 22 chromosomes are numbered 1 through 22, and then we have the sex chromosomes X and Y. And there are two copies of each chromosome 1 through 22. These are called a homologous pair. And a key point here that you need to understand in order to understand genetics is that in general we have two copies of most of our genes on a chromosome. This is a karyogram, which is a picture of the chromosomes inside a human cell, and you can see that there are 23 pairs as we just discussed. Most of the cells in the human body are somatic cells. These cells are referred to as diploid, meaning they contain two sets of chromosomes in 23 pairs like we just saw in the last picture. A small subset of our cells, however, are called gametes. These are reproductive cells, and they are haploid. They have only one set of chromosomes. They're designed to merge with another haploid cell to create a diploid offspring. The somatic cells in our body replicate through a process called mitosis. This involves an S phase of the cell cycle where the chromosomes are replicated into two sister chromatids. Then there is an M phase, which is where mitosis occurs, where the cell divides, and a daughter cell is created with copies of the chromosome. To understand this, I've drawn a picture at the bottom of the screen. So we begin with a somatic cell, which has a homologous chromosome pair, two copies of a given chromosome. For example, let's say this is chromosome number 11. In the S phase of the cell cycle, DNA is replicated and the chromosome becomes two sister chromatids, as shown in this picture here. The cell then divides to become two daughter cells, which contain copies of the original chromosomes. Reproductive cells undergo a process different from mitosis. It's called meiosis. This occurs in gametes. This generates cells that are haploid with one set of chromosomes. It's generated by the process of meiosis of cells called germline cells. And then two haploid cells will merge in fertilization to create a new diploid cells with half of the chromosomes coming from one parent and half from another. So a key point here, which is another thing that comes up again and again in genetics, is that in general offspring get one gene from their mother and one from their father. Some more terminology on this slide. An allele is an alternative form of a gene. It turns out that many genes have several forms. For example, the gene for eye color has multiple different forms, and that's why human beings have different eye colors. The same is true of many other genes. And in genetic textbooks and in genetic descriptions, this is often represented by a letter. For example, the A gene will have one form denoted by a capital A and another form denoted by a lowercase a. When we say a gene displays genetic polymorphism, we mean that it exists in multiple forms and has many different alleles. When we discuss the locus of a gene, the plural is loci, we're talking about the location of the allele on a chromosome. That's what the word locus means. So don't get confused. Remember that our cells contain DNA, which makes up our genes. Genes are found in multiple different forms called alleles. Each allele is located at a particular spot on a chromosome called a locus, and you can find the loci for various genes on particular chromosomes. The genotype is the genetic makeup of a cell or individual, and it often refers to the names of two copies of a gene. For example, if a person inherited gene A from their father and gene B from their mother, that person's genotype would be said to be AB. Or sometimes it refers to two alleles of gene A. For example, if gene A comes in the form big A and the little a form, the person's genotype might be two big A's, one big A and one little a, or two little a's. The phenotype, on the other hand, is the physical characteristics that result from a genotype. For example, genotype AB might equal phenotype blue eyes, and genotype BB might equal phenotype green eyes. The wild-type version of a gene or allele is the type that's common in most individuals. For example, for many genes that come in more than one allele, they will say the capital letter version of the gene, such as capital A, is the wild type. The mutant version of a gene or allele is different from the wild type. It's caused by a mutation, and for example, The lowercase letter often denotes the mutant in genetic examples, so an individual might have genotype capital AA, meaning they have two wild type or normal versions of the gene, or they might be a mixture with one capital A and one lowercase a, or they might have two copies of the mutant version of the allele. When we say a cell or an individual is homozygous, we mean that they have two identical copies of a gene. For example, imagine the A gene comes in two alleles, one we'll call capital A, the other we'll call lowercase a. A homozygous individual could have two copies of the capital A or two copies of the lower a version. A heterozygous individual or cell, on the other hand, has two different copies of the gene, such as capital A and little a. Mutations in our gene are what lead to different alleles and different versions of a particular gene. So let's talk about types of mutations. A germline mutation is a mutation that occurs in the DNA of sperm or egg cells, in other words, of gametes. This type of mutation can be transmitted to offspring, and when it is, the DNA mutation will be found in every cell in the body of that offspring that inherited that germline mutation. Somatic mutations, on the other hand, are acquired during the lifespan of a somatic cell. They are not transmitted to offspring. So for example, if one of your skin cells acquires a mutation, that is never going to be passed on to your offspring. However, it will exist in all the daughter cells of that particular skin cell. A dominant version of a gene or a dominant allele is one that can determine the phenotype even among individuals who have only a single copy. It is often denoted with capital letters. For example, if a given gene has two alleles and one is denoted by capital A and the other by lowercase a, then individuals who have one copy of capital A and one lowercase a, or individuals who have two copies of capital A, will all have the A phenotype. In other words, the capital A allele dominates over the lowercase a allele. In contrast, a recessive gene or allele requires two copies to produce a phenotype. This is often denoted with lowercase letters. For example, only individuals who have two copies of the lowercase a allele will have the lowercase a phenotype. Individuals who have a capital A and lowercase a, or two capital A's, will have the capital A phenotype. Codominance occurs when there are different alleles for a given gene, but none are dominant over the other, and both alleles in an individual contribute to the phenotype. The classic example of codominance is the ABO blood groups. If you have the A gene, then your red cells will have the A antigen on their surface. If you have the B gene, then your red cells will have the B antigen on their surface. And if you have the O gene, your red cells will have neither A nor B. Individuals who are A and B express both the A and B antigens. That is because they are codominant. Neither one dominates over the other and excludes the effects of the other. The rare condition alpha-1 antitrypsin deficiency also displays codominance, and this is often tested on step 1. This condition may cause early COPD and liver disease, and I discussed the clinical aspects in detail in the video in the pulmonary section on obstructive lung disease. But for our purposes here, just know that mutations in the AAT gene, which produces alpha-1 antitrypsin deficiency, lead to this disorder. The M allele is the normal version of the AAT gene. If you have the S allele, you will produce moderately low levels of the alpha-1 antitrypsin protein. If you have the Z allele, you will produce severely reduced protein levels of alpha-1 antitrypsin. So the combination of alleles determines protein levels, and this is because they display codominance. So if an individual is MM, that individual will be normal. If an individual is ZZ, that individual will have a severe deficiency of alpha-1 antitrypsin. Other combinations lead to variable levels of disease depending on how much alpha-1 antitrypsin protein is produced. Penetrance is a genetic term used to describe the proportion of individuals with an allele that express the phenotype for that allele. And there are a number of diseases which display what's known as incomplete penetrance. This means that not all individuals with the disease mutation develop disease. It's often applied to autosomal dominant disorders where you would expect everyone that carries the gene to have disease. However, in disorders that have incomplete penetrance, not all patients with the autosomal dominant disease gene develop disease. And a classic example of genetic mutations that display incomplete penetrance are the BRCA1 and BRCA2 gene mutations. BRCA1 and BRCA2 are genetic mutations that lead to breast cancer. They are germline gene mutations and they are autosomal dominant. However, many women born with these mutations do not develop cancer, and this has important implications because although women that have these gene mutations are at increased risk, they may not develop breast cancer, and therefore if you are going to apply a cancer risk reduction strategy like prophylactic breast surgery, You don't know whether or not it'll benefit the patient since they may not develop cancer because these gene mutations display incomplete penetrance. Expressivity refers to variations in phenotype of an abnormal gene. This is different from penetrance, and this is often a point of confusion. To understand this, let's discuss a classic disorder that displays variable expressivity, and that is neurofibromatosis type 1. This is a neurocutaneous disorder where patients develop brain tumors and characteristic skin findings. It's an autosomal dominant disorder, so you expect all individuals to have disease, and in fact they do all have disease. This disorder has 100% penetrance. However, there's great variability in the disease severity, the number of tumors and the skin findings, and for this reason neurofibromatosis has variable expressivity, although it has 100% penetrance because everyone with the gene does get the disease. Pleiotropy is a term that refers to the fact that one gene or one gene mutation can have multiple phenotypic effects and traits. For example, a single gene mutation could affect the skin, the brains, and the eyes. And there are many examples of disorders which are caused by a single gene mutation yet lead to findings in multiple different organ systems. For example, phenylketonuria or PKU can affect the skin. It leads to a characteristic body odor, and it also causes mental disability. Marfan syndrome leads to findings in the limbs, the eyes, and the blood vessels. Cystic fibrosis can involve the lungs and the pancreas, and osteogenesis imperfecta can involve the bones, eyes, and hearings. And there are many other examples of genes that have pleiotropic effects and genetic disorders that are caused by a single gene mutation but lead to abnormalities in multiple organ systems. For the next few slides, I'm going to discuss the two-hit origin of cancer. This refers to mutations in tumor suppressor genes that can lead to cancer. And the word tumor suppressor gene is very important because the two-hit origin of cancer only applies to mutations in these genes. Tumor suppressor genes are genes with many roles in cells. They are often gatekeepers that regulate cell cycle progression. Sometimes they are DNA repair genes. And if you have a heterozygous mutation for one of your two genes of a tumor suppressor gene, this will lead to no disease. You need to have mutations of both alleles to develop cancer. And for this reason, it's said that cancer requires two hits, meaning an abnormality in both copies of a tumor suppressor gene, or another way this is sometimes stated in genetic textbooks is that you need loss of heterozygosity in order to develop cancer related to a mutation of a tumor suppressor gene. Here's a picture from the web just to make this very clear. A normal individual has two copies of tumor suppressor genes, one found on each copy of a homologous chromosome pair. If they develop a mutation and lose one of the tumor suppressor genes, they still won't get cancer because the other gene will continue to function. This is said to be the first hit. However, if they develop a second hit and lose the other copy of the tumor suppressor gene, they will then have unregulated cell growth in that particular cell that has lost both copies of the tumor suppressor gene, and that will lead to no functional tumor suppressor effect, and that will lead to malignancy.